We'll kick off in the interest of time. You're very welcome to the Mansion House. Thank you very much for coming along. This is goodly numbers this evening. Can everybody hear me okay?
Yes.
Liam and I are just, we feel this is a little bit austere to kind of hide behind. So if our voices carry, we're okay. But if anybody is having problems hearing us, just let us know and we'll move up there. As I said, you're very welcome. My name is Robert O'Connor. I'm the Head of Research for the Irish Cancer Society. And you're here this evening for our latest in our Decoding Cancer talks, Tackling Breast Cancer with More Precision, which will be delivered by Professor William Gallagher. Just a little bit about our Decoding Cancer series. We try in the research department of the Cancer Society to try and better engage around scientific research and facts and information. Our organisation is wholly funded by the public, so we feel we have to, there's an onus on us to engage with the public to better understand what research is doing and the tremendous advances that are taking place and some of the other challenges that exist. I'd like to thank, as I say, the Lord Mayor and his staff for facilitating us in a fantastic venue, very august venue. So we're here to talk about breast cancer, just a little bit of kind of some of the background for the Irish breast cancer situation. This year about 3,000 women will get a diagnosis of breast cancer. About 300 more will get a diagnosis of an earlier stage form of breast cancer called in situ. And breast cancer has seen some tremendous advances in treatment, including in this country in the past few years. So mortality, for example, has halved over the last 20 years. We have seen tremendous advances, but we're moving into the next phase of those advances. And research groups like our Irish Cancer Society funded Breast Predict are really at the cusp of that. And I think that's what one of the things that Liam is going to talk to us about this evening. About 3/4 of breast cancer patients will be cured of their disease. Thankfully, that's the good news. Obviously, the bad news of that is about 1/4 of patients won't be, and they may end up with secondary disease where treatment options really are at the forefront of learning and technology. A little bit about our main speaker. Professor Gallagher is, he's got a few different hats. He's the director of UCD Conway. He's Professor of Cancer Biology in University College Dublin, and he's also director of our Breast Predict programme. And that's a major investment by the Irish Cancer Society. Back in 2014 now, we're in the 2013, we pulled together the best and the brightest in Ireland to come up with their ideas. And Liam led the group that was successful when we got that externally peer-reviewed by international experts with an excellent programme. And we put €7.5 million into that programme to try and better individualised treatment and outcome for women, Irish women with breast cancer. Liam's research mostly focuses on cancer biomarkers. He's some interest in some other areas of cancer in addition to breast cancer. And he also has a number of other interests, particularly in the commercial space. He's the Chief Scientific Officer at a UCD campus spin-out called Oncomark. His CV says he's 181 publications, but I actually suspect that number, it goes up on a kind of a weekly basis almost, so there should probably be a counter somewhere that, another publication come in, and that's just the ones that he's involved in. The larger Breast Predict group are producing a phenomenal amount of scientific output for that investment that we're putting in. He's also, he also has 9 patents. He's had numerous accolades, but just a couple of the more recent ones. He's had he's been awarded the St. Luke's Silver Medal, and also earlier this year, this year in February, he was awarded by his peers in the Irish research community the first and inaugural IACR, Irish Association for Cancer Research Medal, recognising excellence in cancer research and leadership in the Irish space. So, I said you're very welcome again this evening. What we would ask is maybe that Liam go through his presentation. He's very kindly agreed to answer any questions and that at the end. We are recording the presentation because we'd like to try and make these things available for the general public. So as I say, we'll be recording and that. Liam, can I pass over to you?
Yep.
And thanks very much for coming along this evening.
So
firstly, I'd like to thank you for coming, braving the traffic on a Thursday afternoon, evening in Dublin. It was particularly kind of difficult to get in here this afternoon. And what I normally do for these types of talks, I actually was really looking forward to this talk. I normally actually also rent a crowd just in case. So my parents are in front and my daughter, just in case nobody turns up, I always bring a crowd. And I'd like to thank Rob and the other members of the Irish Cancer Society research team really for the support they've given us over the last 4 years, particularly in the case of Breast Predict, and I'll cover Breast Predict a bit later. What I was hoping to do at the start really is kind of just stand back and I suppose kind of give a kind of a few minutes to describe what is cancer and particularly what is breast cancer, and just kind of start from that basis. And the title of my talk is really, how do we've learned, I suppose the summation of it is, we've learned a lot about kind of how breast cancer arises, what are the kind of underlying biological causes to breast cancer. This has resulted in a lot of different treatment options, but ultimately really what we're hoping to do is really move towards a situation where we can actually take each individual patient and treat them accordingly in a very targeted and precise way. And we have some examples of that, which I'll cover, which some historical examples. But really what we're really doing, at least in terms of research that we're involved with, is really trying to identify the key targets that we can actually drug. So first of all, I'd like to dedicate this talk, and this is the first time I've done it, and I've particularly done it because my parents are here, is 3 family members which are affected by cancer. My Aunt Elizabeth died of lung cancer a number of years back. My Aunt Mary, on my father's side, currently has metastatic colon cancer, and my Uncle Eddie on my father's side is a survivor of prostate cancer. So again, this is a picture, I suppose, that a lot of families have. Unfortunately, the statistic was 1 in 3 people would get cancer during their lifetime. This is probably will increase to 1 in 2 individuals will get cancer during their lifetime. Because we're living longer and there's a number of issues associated. That doesn't mean we're going to succumb to the disease, but we're obviously going to experience it throughout. So first of all, what is breast cancer? And more generally, what is cancer? And I think some of the things I'm going to talk about here are relevant to not just breast cancer. And again, when we talk about breast cancer, we're talking about essentially, I suppose, cells which are normal cells within the breast. These are cells which are, so cells are small subunits of, or components of our body. We have hair cells, skin cells, but we also have, for example, within the breast, cells which line a duct, which produce milk, and lobules, which it can actually, due to inherited mutations, or just to living longer on the earth, or exposure to carcinogens, or various dietary factors, something can go wrong. And these cells can change, they can become abnormal, and then they grow beyond control. And so, for example, this is, I suppose, a representation of a breast tumour within the breast. And I suppose one key aspect is early detection in breast cancer, but also trying to understand what's the underlying cause, why has this happened? Because if we can understand why this happened, we can actually do something about it. So this is a picture, a really detailed picture of cancer cells down a microscope, a very essentially high resolution microscope. And these are essentially just a representation of cells. Each one of those kind of blobs is a cancer cell, and some of them, they actually, interestingly, these cancer cells can actually communicate to each other, either by physical contact, but also sending out messages which can actually have effect on their growth. So I'm a cancer biologist, and what I suppose I spent my career doing is trying to understand what's going on inside that cancer cell. What, you know, if you think about it, it's a bit like a clock that doesn't work. You open up the clock, and you find out what are the kind of elements within the clock that aren't working, and try to fix it. So that's what we do as a detailed lab scientist. We try to understand what's going on inside that. And key for me is not just to find out, but actually use this information for the improvement of breast cancer patients. So this cartoon is from a paper which a PhD student in my lab wrote about 10 years ago. And she went back and studied medicine, Sharon McGee. But it's a cartoon, I suppose, representation of what happens. For example, these are individual cancer cells. And like any tissue in our body, these need to be nourished by by the blood supply. And so one of the key steps is really the nourishment of the tumour by the blood supply. And when we talk about primary breast cancer or early breast cancer, we're talking about the tumour being confined to the breast. And as Robbie indicated, in the majority of cases that can be controllable, particularly, and I'll talk about the different forms later. I suppose one of the caveats is that these tumour cells can actually, or these tumour masses can actually release tumour cells into the circulation, into the bloodstream. And these can actually go to different parts of the body, for example, the brain, the liver, to the bone, and actually set up new homes within those sites. They can stay dormant for many years, but then at a certain point it can actually grow and form a secondary disease, or what's called metastasis. And that can be very challenging to treat. And when you're trying to treat breast cancer, commonly if you have primary tumour, the tumour is excised or removed surgically, radiation treatment is applied, maybe chemotherapy. And the chemotherapy is essentially, fundamentally, classic chemotherapy is similar to a kind of general, I suppose, poison to a certain degree. You're trying to poison, kill the tumour cells in circulation and hope it doesn't have an adverse effect on the person. And the idea behind that is to remove these cells, or try to kind of stop these cells setting up site. So I'll come back to this a bit later. And Michael's going to get annoyed with me because I'm going to go up here. But this is actually a movie, and I use it quite a lot. And what I'm just going to show you is this is actually a movie looking down on a model system, an animal model, where you're looking at human tumour cells which have been put into that animal, but a fluorescent tag is attached to those cells. You can actually see. Yeah, so you can see the cells moving along here. So these are tumour cells. This is a blood vessel. You can see these tumour cells rolling along here, and some of them have stuck. See the way they're kind of, it's like a football. And the analogy I have, if you see the floorboards down here, it's like if I had a football and I kicked the football along the floorboards. It bounces, rolls, and at a certain point the football stops and squeezes in between the floorboards. That's what happens with cancer cells. It squeezes out of the floorboards from the circulation to set up these sites of metastases. And I've probably done something really bad here. This other picture is, so that's the entry into the bloodstream. And this is just a picture, it probably won't work, but where you're looking at exit, the squeezing between the floorboards. Hopefully you can see it. These are a clump of cancer cells, a clump of cancer cells here. And I won't show it, but ultimately in the video what you see is the cancer cells are squeezing in between the cracks. These are individual cells lining the blood vessels, and these cancer cells squeeze in between those cracks, in between these blood vessels, and set up a site. Okay, so that's enough of up and down. So when we talk about the spreading of the cancer from the primary site to the secondary site, there seems to be kind of certain areas that, it's particularly breast cancer, they tend to go to. Okay, and that's actually an important research question, because if we can actually identify why a certain tumour might go to the brain, might go to the bone or to the lung, then we can actually combat that very effectively. And that's one of the strands of the research that's taking place in Breast Predict, led by Leonie Young and the College of Surgeons, is try to identify how that works and try to actually intervene. So the actual, I suppose the title of this kind of whole series is about decoding cancer. And I'm a biologist, so I suppose I spend my life trying to understand, fundamental workings of biology. And what it's really about is, I suppose, the genes within our cells. And you've probably heard of DNA. This is the kind of instruction set within our cells. And people worldwide have spent many years and billions of euro and dollars describing what's known as the human genome, describing the kind of catalogue of instructions within DNA. Because when we talk about cancer, it can just take one or two small changes within our DNA to have a very specific effect. It's not just cancer. For example, in the case of cystic fibrosis, it's just one small change in 3 billion letters that causes cystic fibrosis. Same with cancer, there can be one or two small changes which can have an impact. And I suppose what a lot of research has been is trying to identify what are the genes which cause cancer, and actually how does it work, and then can you drug it. And again, when I talk about these letters, this is really the instruction set that's within our DNA. Breast cancer is a growing problem. In Ireland, in terms of females, have a 1 in 10 chance of getting breast cancer. Obviously, breast cancer can also occur in males, but at a much lower incidence. We know that at a worldwide level, breast cancer is increasing in terms of incidence, and this could be down to improved screening, detecting the actual breast cancer earlier, but also lifestyle changes across the world. And also, again, as we get older, we will succumb to more cancers. And interestingly, with the kind of, I suppose, potential eradication of the HIV epidemic in certain parts of Africa, people are living longer, and actually cancer is becoming a problem, particularly in Africa. And actually I was at a meeting in Madrid there about 2 weeks ago, and I was astounded. There's only one medical oncologist in the entire country of Malawi. 17 million people. He actually has 100 patients a day he actually sees. No infrastructure in place to actually treat people for cancer, no access to therapies. So, there's huge kind of inequities across the world in terms of access to particular treatments. So while the news is not all that bad, because ultimately we're detecting the cancer earlier, so mortality is decreasing. We're actually having an impact, particularly in breast cancer, particularly with early detection and better treatments. So, for example, if women or patients present with a localized disease where it's confined to the breast, they have a very good outcome. If the disease is kind of partially spread to kind of adjacent lymph nodes, it tends to be slightly less. Unfortunately, one of the kind of key targets for us is if the disease is spread to different organs around the body, then this is the survival over a period of time can be significantly reduced. But again, this particular area is actually, we're seeing some successes recently. So importantly, most women diagnosed with breast cancer now survive. And this presents a key issue, and this particular area that we work on is it's more important that we actually tailor our treatments and also avoid unnecessary side effects. It's a key issue. Sorry, it didn't come out very clearly, but ultimately we know certain things, some factors are changeable, we can actually maybe do something about, and some things we can't change. Okay, I can't change my parents, okay? They they can't change me, but even if they try. But ultimately, you obviously inherit good and bad things from your parents. Some of that, unfortunately, can be a predisposition for cancer. And I'll come to that particularly in breast cancer. Obviously, we're getting older, so again, the longer we live on Earth, the higher the risk we're going to get cancer. So if we actually develop the magical cure for extending lifespan, unfortunately, unless we address the cancer situation, you're going to get cancer during that time. But there are factors which are changeable. There's obviously data showing very clear links between obesity, poor diet, alcohol, lack of exercise, and hormone exposure in terms of increased risk of breast cancer. And again, it can be quite subtle in terms of those effects, but kind of cumulative effects it can have an impact. There's also this concept of what we term sporadic or random breast cancer, and what we also term familial and hereditary breast cancer. So what I mean by that is on the right-hand side, and I'll give the example of Angelina Jolie. She has a mutation in a gene called BRCA1. And this is a case where you inherit this mutation. Every single cell in your body will have that mutation. A small change, and this BRCA1 gene is a key protector. It's kind of like a guardian gene, and if you have a mutation in that gene, it doesn't mean you're going to get breast cancer. It just you have a very increased risk of getting breast cancer, around 80 to 90% risk of getting breast cancer during your lifetime. And there are other genes like that, not just BRCA1. There's a whole variety of genes which, if there's a mutation in it, gives you a very high risk of getting both breast and maybe also ovarian cancer. And then there's ones which are more subtle, not as strong, so it's a combination of effect. So these are we term familial and hereditary components, which account for about 25%, 1/4 of breast cancer. The other 3/4, it's not every single cell in your body, it's just cells, as I said, which line your duct. So these are kind of, for example, exposure to particular carcinogens, etc. And again, people will be obviously familiar with the concept that Angelina Jolie she had a number of close family members which were diagnosed with and died in some cases from either breast or ovarian cancer. And if in that context, if you have individuals which have a number of relatives, first-degree relatives, which are affected by breast cancer at a young age, there's a good chance or a reasonable chance that you may have this hereditary component. Doesn't mean you do have it, could have a chance. She was tested for this particular mutation, this BRCA1 mutation. Again, she had, at that point, 87% risk of getting breast cancer during lifetime. I think she was 39 at the time, something around before 40. And she decided to get a double mastectomy as a preventative strategy to get against breast cancer. Now in that context, she reduced her risk of getting breast cancer from 87% to 5%. Even though the breasts are removed, there's still a chance that she could get breast cancer. I'm not saying I'm not advocating this, I'm just saying that was her choice. That's her personal choice to do that. I think more recently, actually, she was also diagnosed, I think in 2015, with early stage, what appeared to be ovarian cancer, and she also had her ovaries removed. Now, what was interesting from an awareness, this is Breast Cancer Awareness Month. Given, everybody pretty much knows who Angelina Jolie is, right? And she was highly publicized. This had a huge impact in terms of awareness in terms of breast cancer. There was a huge spike in interest for people, particularly who are potentially at risk, to actually get tested. And then, unfortunately, there was a drop-off. So again, with anything in the media, there's a peak of interest and a drop-off. So I'm going to spend just a bit of time talking a bit about the past, because it's important to recognize the past when we're talking about the future. And I won't go into the detail, but ultimately, over the last 100 years, a lot of really good research has taken place in terms of uncovering, developing different treatment strategies in breast cancer, different therapies, different diagnostics. And I'm going to highlight just 2 or 3 of them just as a context. The first is actually, I did my PhD in Glasgow, and it was named after this guy here called George Beatson. It was called the Beatson Laboratories in Glasgow. And he was a surgeon who published a very key paper in a journal called The Lancet. It's one of the top journals in the field around the turn of the century. And what he noticed, he was one of the key people who provided this connection between the breast and the ovary, the communication between the ovary and the breast. And he came up with a pretty revolutionary strategy, which was you had individuals with breast cancer. Well, how do you actually tackle that? Well, we remove the ovaries. So you actually remove the ovaries of individuals with breast cancer, and in the case reports that he published, there was a therapeutic effect. Now, obviously, that's very radical, surgical removal of the ovary to have a therapeutic effect. But it actually opened then the door for the chemical strategies, particularly in the case of development of tamoxifen. Instead of removing the ovaries, actually using drugs which actually interfere with hormone function. And interestingly, there is an Irish context, and I'll come back to this, is that the drug tamoxifen, which has pretty much saved more lives than any other drug, anti-cancer drug to date, it's been around for a number of decades, was actually the first clinical trial was actually with tamoxifen, was from an unsung hero in Irish cancer research, a female medical doctor from Northern Ireland called Maya Cole, who spent most of her working life in Manchester. This was the actual paper of, I think, 1971 on the first clinical trial, first use of this drug in women. And in this case, these weren't selected women in terms of, and I'll come back to that in a minute. The other person I want to highlight is actually Dennis Lehman. And Dennis Lehman is also a very good friend of Ireland, because, again, I'll come back to one of the most effective treatment strategies for a subset of patients with breast cancer, which are what's called HER2 positive, is the use of drug Herceptin. And Dennis and his colleagues in UCLA were pioneers, in conjunction also with key investigators like Professor John Crown here. One other hero I want to mention was actually the person that actually kick-started my career in cancer research, which is Professor Joe Duffy. And he's not obviously the Joe Duffy that everybody else knows about, but he's the more important Joe Duffy to us in terms of cancer research. And Joe was a biochemist working in St. Vincent's with no protected time to do research. He essentially did it as a hobby. There was nobody asked him to do it, he just did it. And essentially he's probably the most famous cancer researcher in Ireland. I would pretty much kind of evaluate that. Nobody asked him to do it, but he actually, one of the key observations he did was he worked in the area of diagnostics. He actually uncovered a whole new kind of a set of biomarkers, what we call diagnostics, which can be used in breast cancer. And he did a series of studies over 2 decades to actually validate this. Okay, so it's going to push forward, and again, we know that within particularly breast cancer, we have a number of different options, and I'll talk a bit now in a few minutes about kind of different types of breast cancer and where we are. But ultimately, we have obviously chemotherapy, which has its use, but chemotherapy is quite general. As I mentioned, I use the term poison. In some cases, we're essentially killing or affecting the growth of cancer cells and trying to win a game against any adverse effects on the human. But it's non-specific. It's what's called non-targeted generally. And there's an increasing shift towards what are called targeted approaches. And one of the best examples of that would be Herceptin. And there's a whole variety of other drugs which either have been approved, which can be used clinically, or are in development. And this is an important aspect, that there's a whole slew of drugs which are in development, which are at the cusp of being released and being used on a day-to-day clinical basis. But unfortunately, it takes time. The average time it takes for a drug to be developed is traditionally 15 years from discovery to release. People are contracting that and becoming more efficient. It can also be quite expensive, in the order of in excess of $1 billion. The one success story, particularly within Ireland, is the Irish participation in clinical trials. And this is really stemming from the establishment of ICORG, now called Cancer Trials Ireland, originally founded by John Armstrong and John Crown. It pretty much involves all of the oncologists, the cancer treating physicians in the entire country, in a real partnership approach. And what this meant is that Irish patients get early access to very innovative drugs, which obviously could have an impact, much faster than in a lot of countries worldwide. And so, for example, these are some of the agents, and trastuzumab is essentially another word for the drug Herceptin, a whole variety of different agents where Irish patients got early access to these therapies. And if you look, for example, in breast cancer outcomes, if you look at a 5-year survival rate, we've increased it from 70% to 80%, and actually more recently beyond that in the last number of years. And this really coincides with really the opening of a huge amount of trials in the breast cancer space, where a large number of women were offered these very innovative therapeutic options. And again, if you look at the participation of breast cancer patients in clinical trials, we actually, again, as Robert indicated, it's approximately 3,000 women who have breast cancer or diagnosed with breast cancer per year. We have a very good proportion of patients of that group which are participating in clinical trials. And there's huge international evidence suggesting that participation in clinical trials, generally in comparison to other, general standard of care, you have a better outcome. Now, so I'm going to step back a bit and just kind of talk a bit about the fact that we know that breast cancer is not just one disease, but many. That's the first main point. And obviously, individuals who have breast cancer know that because they know what type of breast cancer they have. They have ER-positive breast cancer, or HER2-positive breast cancer, or triple-negative breast cancer. They know. But that's very important to actually recognize that. But even if patients are diagnosed with a particular type of breast cancer, like ER-positive breast cancer, you have patient A and patient B. They look the same age, same type of cancer. Unfortunately, they can have a different response to treatment. And we sometimes don't know why that's the case. And that's part of what we're interested in, is finding out why. So within a patient, we can have differences between the primary tumour and when the disease is spread. But also within the tumour itself, you have these various different kind of subcompartments. So for me, as a biologist, what I'd like to be able to do is actually use the information that I'm tinkering in the lab and actually start using it for useful purposes. And one of the areas that we're quite interested in is understanding, drugs often have an initial good response, but disease sometimes comes back and is resistant to that initial treatment. Can we understand why that has happened and try to actually overcome that? And also, how can we redefine how we can actually treat breast cancer? And I'll give you some examples of what's happening, particularly in Breast Predict. Now I'm just going to just talk a bit more detail about kind of different types of breast cancer. And again, people who have breast cancer will actually be very familiar with this. So if you look at, say, a group of patients which have breast cancer, the majority of those patients will be classified as what's called hormone receptor-positive. Okay, and they generally be recommended some type of hormonal therapy. For example, it could be tamoxifen or some other type of endocrine therapy. There's a subgroup of patients, and unfortunately there is still a subgroup of patients. So in these cases, you have targeted treatment options. For example, endocrine therapy or HER2 targeted therapy. But around 15% of patients which are diagnosed with what's called triple negative breast cancer. And classically, these patients are not offered, or there's no option to offer targeted treatment. And one of the core focuses of Breast Predict is to actually develop new treatment strategies for that subgroup of breast cancer. So if you look at the hormone receptor positive, or I'll use the term ER positive breast cancer, which is the vast majority, as I said, tamoxifen and these types of drugs are very effective. They've had a dramatic impact in terms of survival. Obviously, there are subsets of patients which actually don't, they may initially respond, but then the disease may actually come back and relapse, which we need to deal with. Again, I mentioned myocole. A key critical role is the actual is pathology and the pathologist. The gatekeeper for most therapies is actually not really the oncologist, it's actually the pathologist, because the pathologist looks at cancer down a microscope and makes a determination for what type of treatment you're going to get. Obviously in consultation with the oncologist. And this, for example, is different cancer, these are different breast cancer cells stained for this protein called ER, the estrogen receptor. And you see those brown dots? These are cancer cells which are what's known as positive for the estrogen receptor. And so a pathologist would actually look down a microscope on a piece of tissue and actually count those brown dots. One of the key issues, particularly the fact that, if you think about it, in the context of ER positive breast cancer, we have hormone therapy, which is very effective. The key question is, should you also give chemo as well? We all know about the side effects with chemo in terms of the adverse side effects. Most women who have ER positive early stage breast cancer don't need chemo, but there are a significant subset which could benefit from chemotherapy. And to benefit those patients, traditionally, that small subset of patients which could benefit from that treatment, all of those patients were given chemotherapy because we didn't have tools to segregate those. And so this is the idea of biomarkers, stratification of patients. You have 100 patients. Can we identify those patients which potentially could benefit from chemotherapy versus the ones that have no benefit and do perfectly well with hormone therapy alone? And you could, for example, the general statistics are maybe there's about 30% which have a risk of what's called high recurrence risk versus a low recurrence risk. And there is a test, and again, Irish science has played a key role in this, called Oncotype DX. So Oncotype DX is a kind of fancy new molecular test developed by a company in the US called Genomic Health. It's quite expensive. It's around €3,000 to €4,000 per test. And what you're doing is you're measuring not just one factor, one gene, you're measuring 21 genes together. And what you're doing here is asking that question, can I stratify patients into two, and actually not even two groups, into three groups, a low risk, intermediate risk, or high risk of disease spreading. So it's patients with early stage primary breast cancer, you carry out the test and you predict maybe what's going to happen 10 years down the line. If they're predicted as being low risk, you can avoid chemotherapy. Unfortunately, obviously if they're high risk, you will get chemotherapy. We don't know what to do with the intermediate risk. That's one problem. And one area that we work in my lab is actually try to remove this intermediate risk group. And again, there's a big ongoing study which encompasses, it's called TAILOR-X. It encompasses nearly 1,000 clinical sites worldwide, 11,000 patients. And what's interesting from an Irish point of view is that the Irish hospitals that participated in that and the patients were some of the world's biggest accrual centres for that trial. Also, Irish patients were the first patients in Europe to get, where public patients got access to this particular diagnostic in 2011. And again, this is there's 975 participating sites. These are different hospitals in Ireland: St Vincent's, Galway, Mater, St James's. You can see St Vincent's did particularly well. Second highest worldwide out of 971 clinical sites. The other clinical sites are the world's biggest cancer centres, a lot of them. So, you know, Ireland and Irish patients really punched above their weight in terms of participation in this trial. So that's ER positive breast cancer, where I don't have time to go into all the detail, but there's still a lot of work around the area of resistance. How are we doing on time? Okay. So I'm going to shift to HER2 positive disease, and I'm actually going to ask for a bit of audience participation. Okay, don't be worried. So HER2 positive breast cancer, again, it's around 15% of breast cancers, and again, as I mentioned, it was traditionally a very aggressive subgroup of breast cancer, where if a person was diagnosed with HER2 positive breast cancer, it was bad news. Okay? Now, in certain cases, actually, because of the treatment options which have actually been developed against that particular subgroup of breast cancer, it's actually almost reversed, because you can actually do something about it. So it's an interesting turn of events. And the classic way that this is done where a pathologist and actually determines whether you get HER2 target therapies or not is a piece of the tumour tissue is taken from the person by a surgeon. It's fixed in formalin, that smelly formalin stuff, put in paraffin wax, and then sectioned. Very thin slices are prepared. And then you actually use a stain to actually pick up whether you've got high levels of this HER2 protein or not. And so what you have here is 4 different case cancers, which in this case, each of those kind of circles here are cancer cells. And the brown here is essentially the HER2 protein, which is on the outside of the cancer cells. It's like a ring. It's like a ring of staining around it. And what a pathologist would do is they'd stain individual sections, and they'd actually say, okay, they classify patients. There's no brown staining. There's a small bit of brown staining. There's a bit more brown staining, and there's a lot of brown staining. If you've got a lot of brown staining, then you're given HER2 target therapies. If you have no staining or a bit of staining, you're not given HER2 target therapies. The problem is what happens here, and so there's a kind of ambiguity here. And what happens is if patients have this kind of intermediate kind of staining, they're sent for a secondary assay to do the test. And so remember this. So the terminology is 0+ for nothing, 1+ for a bit of staining, 2+ for a bit more, and 3 is a lot. Okay? So now what we're going to do is actually do a show of hands, and we're going to train you into pathologist and see how it goes. So this is an example of again, on one tumour, all 3 types. A small bit of staining, a bit more staining, and a lot of staining. Okay, so I want to ask people to raise their hands, and they'll go 0, 1, 2, or 3. Okay, so 0, raise your hands. 1, 2, 3. Okay, we're all pathologists. I've trained you already. Okay, now that's an easy one. Correct. Okay, 0, 1, 2, 3. Okay, so there's actually no staining there whatsoever. 0. Okay, now we're going to get a bit trickier. Okay, 0, 1, 2, 3. Okay, the majority was 1. It's a tricky one. Okay. Now, this is a real tricky one. This is what causes people problems. 0, 1, 2, 3. And that's the problem. This is a 2. And again, if a pathologist classified as a 3, you would automatically get HER2 target therapies, but it may be incorrectly described because actually it may be 2 and actually not have the phenomenon. Okay, so anyway, well done. And actually, interestingly, crowd surfing is actually starting to be used for this type of application. I won't have time to go into that. But why is this important? So it's important from the perspective of, again, Dennis Lehman was the US researcher, and actually he's also popularized by the fact that, I'm not sure if you know the singer Harry Connick Jr. So there's a made-for-TV movie based on Dennis Lehman. Now, Dennis is not bad looking, but Harry Connick Jr. is a bit better looking than Dennis. But it's actually about his story. And it's an amazing story. There's a book called HER2, and it's essentially about the story about the development of Herceptin. And essentially, initially the drug company, Genentech, was not interested in the development of Herceptin, even though now it's a blockbuster drug. And there was actually driven really, and I'm actually going to that particular area of the neck of the woods on Saturday, Los Angeles. There was essentially there was a lot of breast cancer advocates within the kind of Hollywood's jet set, which actually raised funding and pushed the agenda to actually develop Herceptin. And Dennis was one of the key people, pioneers, to actually develop this agent, which has actually had a very striking impact. The other person is obviously Professor John Crown, and he's worked very closely with Dennis Lehman in terms of developing and using these types of agents. And for example, particularly in St. Vincent's, there's a large number of women which actually have very, very long-term responses to these types of treatments. And there's a whole range of different drugs which have been developed in this space. So that's the past. Now I'm going to talk about kind of going towards the future, and particularly about Breast Predict. And as Rob mentioned, the person that kind of started the vision, and Rob has taken over that vision, is John Fitzpatrick. John Fitzpatrick was a kind of a very prominent urologist from the Mater Hospital who retired, and then he became one of the first heads of research within the Irish Cancer Society. And he had this concept of, instead of people working in silos, a small group here and a small group here competing against each other in a small country like Ireland, why don't we come together as a group, tackle one or two problems together as a collective? And he unfortunately died during the course of Breast Predict, but he was really, he drove forward this singular vision of integration. And so, as Rob indicated, there was a kind of open call for proposals to this new concept of people to come together, a team-based approach to cancer. It wasn't specifically looking for projects in breast cancer, it's just that we put a proposal around breast cancer. And my idea was to bring people together of all the different talents, the surgeons, the pathologists, the lab people, all the different people, all the specialist skills in one collective towards tackle a small number of key questions in breast cancer. And so we put a proposal together which was approved, and we got very significant funding from the Irish Cancer Society. And again, that obviously comes from the people on the ground funding for the Irish Cancer Society. This kicked off 4 years ago. Very significant funding, 7.5 million euro over 5 years. We're 4 years into the program. It's the first collaborative cancer research centre that was funded, and again the goal was to bring people from different backgrounds. And so the whole team is collectively 50 team members. There's kind of 20 scientists like myself of different background. For example, John Crown, medical oncologist, Michael Curran, a surgeon in Galway. Then you've got more lab people. You've got people from various different backgrounds. And then importantly, you've got people in the labs, the PhD students and the postdoctoral fellows, which are doing the research, but also we have people which are facilitating translation research. It involves 6 academic institutions dotted around Ireland, but also Cancer Trials Ireland, which is very important for us. And again, the whole idea was to bring together a group of cancer researchers to tackle a key problem. We encourage people to share resources, not duplicate resources. We focus on kind of key questions which are relevant to patients. We also organise training, because this is kind of sometimes quite a complicated area. You've got people, you have to understand these various different components fitting in, and also use kind of complementary approaches. So we have, for example, computer guys working alongside people who are more in the kind of working in the kind of clinical side. So I use this slide. My daughter said she finds my talks very boring, so she said I will only go to your talk if I show the Care Bear slide. So this is the Care Bear slide, and I also use it to symbolize what I suppose my vision of Breast Predict, which is people coming together from different backgrounds towards a combined vision. We have 5 main goals within the centre. First is really take on board there's a lot of good work which has taken place, which is the currency in this field is the willingness of people, particularly patients, to provide materials for analysis, particularly their cancer tissue, because if we don't have access to that material, we can't actually study and understand what's going on within that cancer. And so a big focus of the centre is on biobanking, collection of blood and serum and tissue from cancer patients throughout their treatment course, and then trying to identify what's going on. And then we have 3 questions which are really fundamentally trying to understand why tumours respond or don't respond to treatment. Can we come up with new combination strategies? One interesting question is women with breast cancer have taken prior drugs like aspirins and statins. What impact does that have on their breast cancer? And also we have a diagnostic component. So this has been a fairly successful effort. And these are just kind of some kind of top line messages from that. Some of these which will be relevant to myself in the science area, but more generally. So we got 7.5 million from the Irish Cancer Society, and one kind of key idea was, because we joined as a group, we were also able to go out there and actually, importantly, if we came up with a new idea, we could actually get funding to actually push that forward and execute it. So using that funding from the Irish Cancer Society, we've been able to leverage an additional 40 million euro from European and other sources and industry to actually progress our ideas.